Okay, so one of the important things we want to examine about this bacteria is its pathogenesis, which is the development of a disease. How does the disease progress? How does it work? So there's various pathways that this bacteria uses. So first, let's talk about attachment. Here you can see the flagellum, which usually functions in movement, but here there's an attachment site and it allows a bacteria to attach to its host cell, in this case this epithelial cell right here. And this attachment then codes or allows for secretion systems, and one of the important secretion systems in this case is the type 3 secretion system. And as you can see here, there's two pathways. First, let's talk about this pathway right here. So, exo-S is secreted into the cell, and what this does is it activates RAS, which is different from the RAS we learned about in previous chapters where in the cell signaling pathways. This RAS is a kinase inhibitor. So let's recall what a kinase is. A kinase transfers a phosphate from one molecule to another. This RAS here inhibits the phosphorylation of ADP to become ATP. And this inhibition eventually leads to disruptive actin in the cytoskeleton. So actin is a component of the microfilaments in the cytoskeleton, and the ADP not being phosphorylized causes the actin in the cell to break apart. And another important pathway here is the exo-U being secreted into the cell, except there's still a lot of research going into this pathway and they're not exactly sure how it affects the cell, and they don't know exactly what goes on, because you can see the exo-U binds to a protein or a molecule over here and plays some sort of role in type 5 and type 6 secretion, but you can see the question mark here. They don't really know yet exactly how it works. And the last thing on this slide would be CFTR. So if you remember what this is, I'll give you bonus points. But CFTR is the cystic fibrosis element of our bacteria. So an interesting fact is that 80% of cystic fibrosis patients are diagnosed with this bacteria, which causes the buildup in their lungs. If you recall, that's one of their main symptoms, the buildup in their lungs of the fluid, and they get infected with this bacteria. And 80% of cystic fibrosis patients get infected with Pseudomonas aeruginosa. Let's move on now to this bacteria's resistance. So, how did Pseudomonas aeruginosa gain resistance? So, one way could be horizontal gene transfer. So, if you can recall back to chapter 18, there were three ways: transduction, conjugation, and transformation. Our bacteria uses transduction and conjugation as seen here. So let's review conjugation. Conjugation requires direct contact between a donor and a recipient cell, and a donor cell transfers a strand of DNA to the recipient. So cell A may have provided cell B. Cell A was resistant to this bacteria, and it transferred the piece of a gene to cell B, and therefore transferring the resistance. The other pathway, transduction, uses a virus. So when a donor cell is infected by a virus, the genetic information, the DNA, the chromosomes, break up into fragments, and on occasion a fragment of that DNA will be incorporated into a newly made virus particle. And then this virus will infect a recipient cell, with that DNA, which encodes for resistance. So that's pretty cool, right? It's helping the cell. And then a natural way, or a way, it's not even natural, it's caused by us. We overuse antibiotics because we didn't have all the knowledge we could have had. And this overuse of antibiotics, essentially, when you're using antibiotics, you're killing all the non-resistant Pseudomonas aeruginosa cells. So when these bacterial cells die, all that's left are the resistant cells. And these resistant cells, well, they survived, and survival of the fittest, they're going to reproduce, and the new cells will have that resistance. So that's why Pseudomonas aeruginosa has a low antimicrobial susceptibility. There's not too many effective treatment options for people infected with this. And another reason of resistance would be these efflux pumps, which I'm going to continue to describe on the next few slides, which use active transport to literally ship the bacteria, the antibiotics right out of the cell. So you can see in this picture right here, the antibiotics come in and whoop, right back out. And this picture actually goes above and beyond because it shows how an inhibitor could eventually lead to a really good, an inhibitor would help these antibiotics stay in the cell and then therefore treat this bacteria. Ooh, I wonder if you heard that lightning. So here's a few different pathways that antimicrobial resistance occurs. So here you can see the antibiotic efflux pump. The antibiotic entered, there was a protein over here, pumped it right out of the cell. There's antibiotic resistance genes, which we talked about, and these could encode for an antibiotic altering enzyme or an antibiotic degrading enzyme. But in the case of our bacteria, the main resistor of antibiotics is this efflux pump. So let's learn more about it. So here's a neat video which shows the efflux pump in action. And this is going to be the end of this section on pathogenesis and antimicrobial resistance of Pseudomonas aeruginosa.